So good evening to all of you. Yes, good evening to all of you. So my voice is audible to all of you. Please let me know, all of you. My voice is audible. Right now you are able to listen to me. All of you, please let me know. Yes, yes. Good evening, good evening, good evening to all of you. Actually, yes, I came little bit early because I have to finish yesterday's topic also. Yesterday we don't have any class. So that's why I think we will have to start little bit early. Yes, yes, all to all, Sapnil, Shubhrit, Ankita, Shruti, all of you, Neha, Megha, Muskan. So you all are welcome. And I have to cover cell culture techniques today. Let me show you the syllabus. I have to complete cell culture techniques today, which we supposed to complete yesterday, but because of so much of network problem, I am not able to complete it. Right? So I will decide that I will complete it today. Along with, I want to start some another topic also. So I will start with cell culture techniques. If everything will be fine, the network issues will not be there, then definitely I will keep moving today, so that we are able to cover our all syllabus on time. Okay, and don't worry, sometime what happens, I am not able to take full lecture, or because of a network issue, because of some problem, you can say because of any reason. Sometimes what happens, I am not able to take your full lecture, so don't worry regarding it. Yes, good evening to all. So don't worry regarding the timing. I will complete your whole syllabus definitely within these 20 days, and I will have to complete all the art which is promised there in the syllabus. So no worries, no issues. So I am now just going to start with the cell culture techniques, the remaining things in the cell culture techniques, because we have to do lots of topics. So I hope you all will getting me properly. So this PPT is visible to all of you. Please let me know. Yes, yes. Good evening to all. Yes, yes. Now I'm just going to start the lecture because we are already very late. Those who will join, after some time, they will watch the recording. So I am now just going to start because, yeah, okay. So I'm now just going to start the lecture because we are very late. I will share the PPT, I will share everything. Let me complete it, then I will immediately if it will finish today, immediately after the completion, I will send it in the Telegram. Don't worry. Right after this class, I will send it. Okay. So now I am going to start with the cell culture. So yes, there are some things which we have to revise again, that we have done only the basics, the very basic things which are there in the cell culture. What is cell culture? And these are all the things which we have discussed in the previous lecture, the applications of cell culture, then the limitations of the cell culture, the things which we have to keep in mind during cell culture. So we have done all these things. Now I am just focused on terminology. Now what is organ culture? This is where we have left yesterday. What is organ culture basically? So this is a three-dimensional culture of undisaggregated tissue retaining some or all the features of a tissue in a vivo. So when organ culture is basically when we are taking a group of cells, when we are working on a tissues or tissue part basically, then it is known as organ culture. Now what is cell culture basically? Cell culture is single cells. When we are working on a particular cell, when we want to know the working of a particular cell, it is known as single cell. No longer no longer organized as tissues. So in tissues always the group of cells they work together, but here there are only single cells derived from dispersed cells taken from the original tissue. So this is the cell culture in which we are working on a particular cell. Now what is primary cell culture? What is primary cell culture? In it it is derived from an explant directly from the animal. So what happened here, we are taking it directly from the animal, usually only survive for a finite period of time. So when we are taking the cells, or we are taking the group of cells directly from the animal, then these cells, the chances are that the cells will die immediately, or they will survive only for a finite period of time. So they will survive only for a finite period of time, involves enzymatic or mechanical disruption of the tissue and some selection steps to isolate the cells of interest. So during primary cell culture, what happens in primary cell culture, we take the cells directly from the animal and we want to let them survive for a finite period of time. For that purpose, we have to isolate specific cells of our own point of interest. So we have to do some enzymatic reactions also in primary cell culture, because we want to take our own cells, specifically our own cells. So this is the terminology which is there in the cell culture. Up to now, is it clear? Everything is fine? Let me check it only once. Everything is fine? Yeah, it's fine. So you are able to see the PPT. Yes, okay. Now after it we have, this is the basic terminology which we have discussed. After it we have some safety levels. We have to take some precautions also when we are working in the cell culture lab, as we have already discussed in the very first lecture, that when we want to do cell culture techniques, we want to work along with the cell culture techniques, we have to take care of some things, some precautions are there. Like they are saying, we have to avoid those substances which are hazardous in our to our health. So that substance which causes cancer, like carcinogenic substances, the substance that cause damage to the developing fetus, which is known as teratogen or teratogenic substances, a substance that can cause a mutation in the genetic material that can be passed to the next generation, which is known as mutagen, or you can say mutagenesis. So we have three terms. We have carcinogenesis, we have teratogenesis, and we have mutagenesis. Getting my point? So we have to take care of all these things. So these are the safety measures which we have to take during the cell culture labs. These are the safety measures which we have to take. So like one, the cell culture area where we are working, there should not be, you have to bring your friends, you don't eat any drink or smoke, or even you do not use a mobile phone there. Getting my point? So we are not able to do these things in the cell culture areas. So do not bring your friends, do not eat, drink or smoke, do not use a mobile phone in the cell culture labs, do wear a lab coat. So these are the simple safety measures which we have to take place during our own simple lab experiments also. Then these are the some basic equipments which are there in the cell culture lab. So in the cell culture lab, the very first thing which you will see is the laminar flow. I hope you all will be aware of this laminar flow. What is laminar flow and how to work in the laminar flow? If you are not able, then I will show you one video also. So in horizontal laminar flow, what happens? These provide the most sterile environment for the cells but offer no protection to the operator. So if there is some, maybe I have one. Let me show you if I have the, like this is the laminar flow in which the lady is working. So this laminar flow, it provides the proper isolated environment to the to the cell culture. But of this lab or this laminar flow also contains the UV light, so we have to keep the UV light on for 50 minutes. After it we have to take the work on. So the very first equipment which we need in cell culture lab is the laminar flow. So these provide the most sterile environment for the cells but offer no protection to the operator. Filtered air enters at the back of the cabinet and is directed to the front directly at the operator. So directly at the operator, the air will strike. Then another thing which we need in this laminar flow is centrifuge. We need to do lots of centrifugation during this cell culture. We have to take human derived cells must be centrifuged in the sealed rotors. So these all things are needed in a cell culture lab. Then contamination of cell culture. This is the most important lack of cell culture techniques, that microorganisms they grow very fast than the mammalian cells. So the microorganisms which is basically the contamination, they grow much fast as compared to the culture, as compared to the cells which we want to grow, right? So they grow 10 to 50 times faster than the mammalian cells, which takes 18 to 16 hours to divide. So these microorganisms they grow much faster as compared to the uh, culture or the mammalian cells which we want to isolate. So they are more tolerant to variations in temperature, pH and nutrient supplies than the cells. So cells are most vulnerable to contamination when our aseptic technique is bad and the culture becomes infected with bugs. So basically the mammalian cell or the cells which we want to grow, their growth rate is very slow and they are more prone to infection. So these microorganisms they are able to grow much fast as compared to the mammalian cells. So this is the isolation of the cell line for in vitro culture, that how we are able to isolate particular group of cells or even a single cell line by in vitro. So we have to take the tissue first. Tissue is basically what? Tissue is the group of cells. So we have to take these tissue or cells in vitro. Then we have to make primary culture. From the primary culture, we have to make secondary culture. From this secondary culture, we are able to get the cell lines. Some of you has asked me about the cell lines also. So from the cell secondary culture, by doing processing and processing by enzymes, by digestion, and by removing the unwanted substances, we will able to get the cell lines. Now these cell lines they will be taken further to make single cell isolation. That means if we want to make we want to work on single cell, then we will be able to make the clonal cell lines. Getting my point? So these cell lines, these are our most important step. From here we will be able to get single cells. We are able to get transformed cell lines. In transformed cell lines, the cell lost his own cell growth. So the cell lost growth. So they are immortalized cells basically. And then some cells will die also. But our main focus will remain on these single cells from the cell lines. From the cell lines, we are able to get the single cell. We are able to get the cells but which lost their growth, and also we are able to get the cells which will die soon. So this is the isolation of the cell lines for in vitro culture. Now we have types of cell culture in vitro. We have types of cell culture. First we have the primary culture. So keep in mind, we have the primary culture, we have the secondary culture, and we have the cell lines. So these three steps are quite important. So we will start with the primary culture. Let me check everything is fine. So are you able to get it? I'll yes, I will show you everything. So are you able to get it? Please let me know. Let me complete the PPT. All of you will get your answers soon. Let me complete the PPT because this is only the introduction part only. When we will proceed, you will get the answer of each and everything. Okay. So don't worry. Let me complete it, then I will able to answer your all queries. I will clear the doubts, but let me complete the PPT because some of your doubts will clear automatically. Some of your doubts will clear automatically when we complete the presentation. So just be patient and keep your questions in your mind, and I will answer all the things. Okay, don't worry. So just I am now moving to the PPT. I just came back to see whether everything is fine. So now I am just going to start the PPT again. So yes. Okay, so we have to focus on these main steps. We have primary culture, we have secondary culture, and we have cell lines. So cell lines are quite important because from the cell lines we are able to get our cells of point of interest. We are able to get those cells which lost their growth. We are able to get those cells which will die soon. Getting my point? So we have to focus on primary culture, secondary culture, and cell lines. Now what is basically primary culture, or what are the types of cell culture which are there in vitro? So in primary culture, as we discussed before also, they are derived directly from the animal tissue. Okay, so the group of cells, they are directly taken from the animal tissue, or maybe from the fetus, maybe from the embryo, or maybe from the normal cells, or maybe from the carcinogenic cells. Okay, because we want to know the activity of particular group of cells, so we have taken that particular group of cell either from the animal tissue, either from the embryo, either from the adult, either from the cancer cells, because we are interested in a specific group of cells, so which are known as primary culture. So they are derived directly from the animal tissue, cultured either as tissue explants or as single cells. So when we have group of tissue, we have group of cells, either we can move forward with these tissues, or we will take our specific cells. Getting my point? Either we can move with the group of cells, or we want to do it with the specific cell, or you can say single cell. So initially heterogeneous, become overpopulated with fibroblasts. So basically these tissues or group of cells, they are heterogeneous, become overpopulated with fibroblasts, and these group of cells, they have finite lifespan. So these group of cells, or these cells which we have taken from the animal tissues, they are able to survive in a outside environment for a finite lifespan. Like we have did the history also, that some scientist has taken the embryo cells or the thick embryo cells in the saline solution. So the primary culture, they will be there in the finite lifespan. They retain differentiated phenotype. They mainly encourage dependent, and they exhibit contact inhibition. So these are some little bit points regarding the primary culture. What happens in secondary culture now? Secondary culture is obviously derived from a primary cell culture. So secondary culture is derived from primary cell culture isolated by selection or cloning. Now here what happens? We will become more specific. Like in primary culture we have lots of different types of cell, but here in the secondary culture we will become more specific. So becomes a more homogeneous cell population, right? Like here there is a heterogeneous cell population, but here it becomes homogeneous cell population. Okay, another things will remain same. finite lifespan, retain the differentiated phenotype, exhibit contact inhibition. The other points will remain same, but yes, there is one change, that from heterogeneous it becomes homogeneous. So that means secondary culture becomes more specified. So after it we have another term which is there, which is continuous culture. So after it we have another types of cells which is known as continuous cultures. Now what happens in continuous cultures? They are derived from primary culture or from the secondary culture. Basically they will be derived from the secondary culture, which is more precise in nature. And what happen here? They are immortalized. That means they are spontaneously or by transformation vectors they are immortalized. They serially propagated in the culture, showing an increased growth rate. So here in continuous culture we have increased growth rate of the cells. So in the continuous culture the cells are keep growing, and definitely they will have homogeneous cell population. Instead of heterogeneous they will have the homogeneous cell population. The loss of encouraged dependency and contact inhibition, but here there is a loss of encouraged dependency and contact inhibition. They have infinite lifespan. Like here we have finite lifespan, but here in case of continuous culture we are able to grow them as infinite lifespan. Okay, so they are the continuous cultures, but the cells which are there in this continuous culture, they are genetically unstable. They are genetically unstable because we are growing it in a outside the living organism. So these are the types of cells which are there. These are the cell morphologies vary depending on the cell type. We have fibroblastic cells, we have endothelial cells, we have neuronal cells, we have epithelial cells. These are all the cells of the our body part. They are present. The neuronal cells are there for, which are important part of the nervous system and everything. So we have different types of cells. We have different types of working of all these cells. Epithelial cells have different working or different functions. Endothelial cells have different functions. So these are the cell morphologies or the types of cell on which we are taking for the cell culture. Either we will work on fibroblastic cells or we will work on neuronal cells, depend upon the type of cell which we want to study. So these are the types of tissue culture. We have primary tissue culture, we have secondary tissue culture, and we have continuous tissue culture. We have already discussed primary culture, which is the, which are basic culture which we have taken from the animal tissue directly. So they are the normal cells which are cultured without any change in their division rate. So they are the normal cells. They will keep growing as they have their own functioning inside the body. In continuous we have two types. We have finite and we have indefinite, or you can say we have finite rate or infinite. Like here we are saying that we have infinite lifespan. So in finite, single cell type roughly 30 times of division enhanced by growth factor. So in finite, the cell will grow but enhanced by some growth factors, some nutrients which will be provided from the outside for these type of development. Otherwise the cell will die. But if we want the cell to divide little bit extra, we will provide them the growth factors outside. Now what happened in indefinite? It is nearly the same as the finite, but the cells here can divide indefinitely. So the growth rate is without any control. So they can divide indefinitely by transformation into tumor cells. So they are also called cell line. So this is the answer lots of you are asking. So they are cell line. So in continuous culture we have two types. Now these are the characteristics of the continuous culture. But when the continuous culture is growing for indefinite period of lifespan, or here the cells are keep growing and growing, and it is growing and growing indefinitely by transformation into tumor cells. They are called as cell lines. So we have just started from the primary culture, and we have to keep moving up to cell lines. So you must know the difference between all these three. So this is the cell line. So these again the same definition. We have main types of cell culture according to the cultivation period or survival in a culture. That how long the cell will survive in a culture. It is divided into three types, or the following types. Number one is primary culture, which we have done. That they will have the normal growth. Continuous culture comprised of a single cell type, or you can say single cell lines. When we are working on a single cell type, it is known as cell lines. And cell line is further of two types, like normal growth and a still divide for a finite lifetime, and they will die after 30 divisions. Normal diploid cells, they maintain some degree of differentiation. They have finite life and they will die after 30 divisions. But in case of indefinite cell lines, in case of indefinite or infinite cell lines, like tumor cells, they transform cells, they will keep growing. Like aneuploid number of chromosomes, they limit span. So they have unlimited lifespan. However, the other two have limited span, but they have the unlimited lifespan. So like suppose this is the developmental stages. This is the group of cells in the morula stage. From the blastocyst we have taken the group of cells. We have inner cell mass. From these inner cell mass we are able to know the functioning of every group of cells. If we will isolate these muscle cells, then we will keep growing these cells to know the proper functioning of that particular group of cells. Getting my point? So these are the simple terms which are there in the cell culture lines. Let me check everything is fine. Then I will pursue. Okay. Is it fine? Everything is you are able to understand everything? Audible? I have to finish this little bit fast. I have to finish it today, so that is why I am little bit fast, because we are not able to complete it yesterday. So I am sorry if you are, if I am little bit fast, and I am really sorry. But I think you will be able to get it properly. Okay. So I am just doing it little bit fast because we have to cover the cell culture lines, and even we have to start the another topic also. Okay. So after it we have, yes, after it we have stem cells. You must be aware of stem cells, but I have to cover these all things. What is basically stem cells? They have the ability to self-replicate. They have the ability to self-replicate and also differentiate to a multitude of different cell types needed within the body. So stem cells are the basic cells which have the ability to replicate as much as possible, and also they differentiate into multiple cell types. So from a single cell we will able to grow RBC cells, we will able to grow skeletal muscle cells, cardiac muscle cells. From a single cell we are able to grow so much of cells which have different functions. Like skin cells have their own function, thyroid cells have their own function, Lung cells have their own function, but they all derived from a single cell, which is basically the stem cells. So stem cells have the ability to self-renew. That means they are able to grow its own self, and also they have the ability of differentiation. So what is basically differentiation? When a single cell carries so much different functions. So this group of cell or this cell is able to work as RBC also, skeletal muscle cell also, cardiac muscle cell also. But yes, this cell get differentiated, so the work gets divided in all these cells. However, all cells are same, but according to the functioning, according to the working, they get differentiated. So this is known as stem cell. What is basically the stem cell? When the cell has the ability to replicate as well as has the ability to differentiate into different cell types is known as stem cell. So the another term which we have to discuss, so we have stem cells which we have discussed right now, and then after it we have hematopoiesis. Now what is hematopoiesis? Basically it is again the same thing. Like in stem cells we have the ability to replicate and we have the ability to differentiate. What happens here? Now this is the basic stem cell. From this stem cell it gets differentiated into B cell and T cell. The basic pluripotent stem cell is there, which gets differentiated into B cell and T cell. This is also the example of stem cells basically, that we are able to get RBCs. From a pluripotent cells we are able to get monocytes, neutrophil, basophil and eosinophil from a pluripotent stem cell. So stem cells are quite important these days. Not these days, actually it's a recent work. Lots of scholars are pursuing their research on these stem cells, that how they have the ability to replicate, how these cells have the ability to differentiate. That means how the power came in all these those cells to get replicate, or not only to replicate but to get differentiate into so much different types of cells. So from a single pluripotent cell we are able to get B cell, we are able to get T cell, we are able to get monocytes, neutrophils, basophils, eosinophils. This is the beauty of, you can say, a group beauty of the cells basically, the pluripotent stem cells basically. So this is the stem cells which has the ability to replicate and which has also the ability to differentiate into as much as types of cells as possible. Now this is a cell cycle. I will not I am not want to discuss this cell cycle. You all must aware of this, that in the cell cycle there are four phases. We have G1 phase, S phase, G2 phase and M phase. So G1 phase is the resting phase basically. So this is the resting state, and S phase is for the synthesis of the DNA, because DNA is needed later on. So this is the DNA synthesis stage. At the G2 phase we have DNA replication, and at the M phase we have meiosis, not meiosis basically, but at the M phase we have mitosis which takes place. So this is whole cell cycle which by which the cell is able to divide. Okay, so this is again the graph of degree of transformation by using the enzymes. So now after knowing the very basic terms which are there in the culture, in the tissue culture, now we have to discuss what are the materials which are needed for the cell growth. You have taken the group of cells. You are interested in a, suppose you are interested in a muscle cells. You have taken that muscle cells as a primary culture. Now what are the things, what are the materials, what is the medium which is needed for the cell growth, how the cell will grow in the outside environment now. So what are the things which are we needed? We need a substrate or a liquid, we need some nutrients which are known as culture media, we have to take care of the environment, we have to take care of the temperature, the humidity, the CO2, everything we have to take care of during cell culture. And the most important thing is to maintain the sterility, to maintain the hygienicity, to maintain the aseptic, to maintain everything contamination free. So this is the main thing which is quite needed in the cell culture lab. So this is the cell culture environment which is provided in vitro. So we have basal media, we have to maintain the pH, we have to maintain the osmolarity, we have to provide the nutrients and the energy source also. So some type of inorganic salts are also needed, like inorganic salts are there for to maintain the osmolarity, to regulate the membrane potential, okay, for the ions, for the cell attachment and enzyme cofactors. So we need these types of media for the cell culture lab. We have to maintain the pH also. We have to need the buffers. The buffers which are used in cell culture lab, they are known as bicarbonate. Bicarbonate buffered media requires CO2 atmosphere, or along with bicarbonate we need HEPES. They are the strong chemical buffer with the range of pH 7.2 to 7.6. Also we need a glucose as an energy source. So these are the things which are required in the cell culture lab. We have to maintain the pH osmolarity, we have to need some growth factors also, we need some media, we need inorganic salts, we need pH indicator, we need buffers. So all these things are needed even in the usual labs also. So these things are quite important in the cell culture lab. Then these are some components of basal media like keto acids, which is oxaloacetate, and pyruvate, which are quite needed during cell metabolism. When we want to grow the cell, so we need these type of keto acids, we need carbohydrates, we have to provide glucose and galactose, we need some vitamins also, because you know vitamins are the precursors for many cofactors. So vitamins are quite important. Why vitamins are important, or you can say why all these things are important? When we want to grow our cell, when we want to keep them growing for long period or for infinite period, we have to need all those things, okay. Even along with it, we need some trace elements like we need zinc, we need copper, we need selenium, and we need tricarboxy acid in the tricarboxylic acid intermediates also. So why all these things are needed? Because these things are there for our normal growth, okay. We need carbs, vitamins, and these keto acids. So if we want our cells to grow, all these type of media should be provided, but from the outside. So all these media will be provided from the outside. Then these are the supplements. The supplements, like we are also taking the supplements these days, because lots of essential nutrients are not in our diet. Our diet is not as good as it should be, so we are taking multivitamins, and we are taking supplements from the outside to keep us growing. Similarly, for the cell culture also, we need some supplements like L-glutamine. Okay, we need non-essential amino acids, we need growth factors and hormones, and we need antibiotics and antimycotics like penicillin, streptomycin, gentamicin, amphotericin B. So we all these things are, you know, quite important in cell culture lab. So we have to provide the supplements from the outside like L-glutamine, non-essential amino acids, then these growth factors, so that the cells will able to grow, the cells will able to maintain the differentiation. What do you mean by differentiation? When a single cell or when a pluripotent stem cell is able to differentiate into red blood cells, into neutrophils, basophils, eosinophils, or you can say it get differentiated into liver cells, muscle cells, lung cells. So this is known as differentiation. So for those all things we need a growth factors, and we need antibiotics also to reduce the risk of bacterial or fungal infections, or to reduce the risk of contamination. Okay, so for all those things we need a supplements. Then this is the medium, like fetal calf or bovine serum, also known as FCS and FBS. All of you have must use these solutions in your own lab also. So these are needed for growth factor, for cell attachment, for binds and neutralize toxins. For all this we need this FCS and FBS, okay. So Then another thing which is need is heat inactivation. Now what is heat inactivation? So here for 50 degrees Celsius for 30 minutes heat inactivation is needed. Now what is basically it? It leads to destruction of complement and immunoglobulins or destruction of some viruses which are there. For all these things we need some heat inactivation. One more thing which we have to keep in mind, the very important thing is that we have to take these growth factors, hormones and vitamins in limit. Okay, so we have to take all those things in limit. If we are taking all those things more than desire, more than need, then it will affect the cell growth. So it will affect the cell growth. So we have to take all those things in the required amount, not more than it. So this is how the cell culture, how we are able to do the cell culture in the lab. I have a beautiful video of this. I will share you later. Okay, so I will share you how to do the, yeah, how do we culture the cells in the laboratory, how to do this. This is only simple diagrams only, but I have a one video regarding the cell culture lab. I will share the link in the telegram so that you will be able to watch it by your own self. So this is the laminar flow on which the lady is working. So the very first important thing is that your laminar flow should be contamination free. Okay, then we have to take the we have to isolate the cells from a group of tissues or group of cells. Then we have to maintain in culture, or you can say in aseptic techniques, we have to maintain that um, the cells which we have extracted outside. After it we have to do subculture, like we have to do primary culture, secondary culture up to cell lines. And after it we have to do the cryopreservation. Cryopreservation is basically what? When we are just storing the cells, the desired cells, at a very low temperature, or you can say at a -degree Celsius temperature. So this is the freeze cells basically. Then we have another term which is there, which is known as passaging cells. Now, I am only just giving you the brief, a simple introduction or simple basics which are there in the cell culture techniques. Otherwise, this field is quite vast. Okay, so if I am unable to cover any topic, please don't mind. But there are only, I am just letting to know the terms which are there in the cell culture techniques. So we have another term which is passaging cells. What are basically these? These cells are here to maintain the cells in a culture, that is, they don't overgrow, to increase the cell number before experiments or storage. So basically what happened here, we want to maintain the cell culture. We don't want that cells will grow more than they are needed. So we have to keep them up to 7 to 80%. Okay, so wash in PBS to remove dead cells and serum. Trypsin digests protein surface interaction to release cells. So when we are able to do the primary culture, when we are able to extract the tissue from the animals, then we have to wash in PBS to remove dead cells and serum. Then we have to do the digestion by the enzyme which is trypsin. Trypsin will help in the digestion of protein surface interaction. Then we have EDTA. This is again another salt. So we have EDTA also, which enhance trypsin activity. So this EDTA is there along with trypsin to enhance the activity of trypsin. We always wash this with PBS solution. Then what happened after enhancing the activity? We have to resuspend in serum. After by one, when the trypsin has done the digestion of the enzymes, then we have to resuspend in the serum containing the media. Transfer dilute cell suspension to a new flask. After it we have to transfer it into a new flask. So most cell lines will adhere in approximately 3 to 4 hours. So you have to keep transferring these culture cells in the different flasks. Now this is cryopreservation of cells. So that is why it is known as passaging cells. passaging means that they keep passing from one step to another step, and they keep passing the flasks also. Then we have cryopreservation of cells. The cryopreservation is basically when we are dealing it at minus or in negative temperature. Like here, when we are able to store these cells at minus 80 degrees Celsius. So we have to keep them at we have to freeze them at minus 80 degrees Celsius, or even we can use liquid nitrogen also, in which the temperature is very low. I think, yeah. So the temperature we have to freeze at minus 180 degrees Celsius, or we can also use liquid nitrogen where the temperature is in minus 196 degrees Celsius. So these are the simple terms which are there in the cell culture. You must aware of this manual cell count, also known as hemocytometer, which is there for the calculation of a group of cells, or even for a particular cell count. So this is basically hemocytometer is there for the cell count only. So this is the how we are able to count the cells. These are the chances of contamination. You all know what is contamination: when there are unwanted or undesirable substances along with our desirable cells. They may be bacteria, yeast, virus, mycoplasm, anything it will be. Or the contamination will also be occur in media, incubator, serum, or even in water. So contamination, the chances of contamination are always there. No issues at all. The chances of contamination are always there. These are some examples of common cell lines which on which the lots of students are working right now. Like we have human cell lines, we have MCF7, we have HL60 cell lines which is for leukemia, we have HELA. Many students are working on it also. So this is basically all cell culture. Now this is organ culture. Now what is organ culture? When we want to grow or when we want to work on organ, specific organ basically. So this is the place where the organs are growing artificially. So if you are able to see, rather than working on a cell, we are growing the organs artificially. Okay. These are again the some basic equipment which are needed in the cell culture labs. Like we need, as I already told you before also, we need the very basic things. Just give me a minute. Like we need laminar flow, we need incubator, we need water bath. So all these things are very basic things which are needed. 1 minute please. So water bath, incubator, centrifuge, refrigerator, cell counter which is hemocytometer, all these things are quite needed. Yes. So all these things, the cell counter, the microscope, the liquid nitrogen chamber, all these things are needed. Okay. I already told you this before also, that there are some basic equipment which are needed in the cell culture labs. We have laminar flow, we have incubator, water bath, centrifuge, refrigerator, cell counter, microscopes and sterilizer definitely. So this is the laminar flow in which I show you here also. This one. So this is the laminar flow. This is the apparatus, how we have to take the reagents, how we have to place the pipette, where we have to deposit the waste. Like this is the waste container in which we will add our waste. This is the ethanol. These are the culture flasks on which we have to work. So I hope you must have worked on laminar flow before also. Okay, so Yeah. So I hope you all have done this on your own labs also. Laminar flow hood is there. Then this is an incubator. Incubator is basically for what purpose? Incubator is for maintaining the temperature. Okay, so we have to control the temperature, or we have to keep the temperature at a constant rate. For that we have an incubator. Then we have backup temperature controller. We have CO2 detector in the incubators. So all these are instruments only. So this is a CO2 incubator design. Then this is the cell culture media. Media. It is of two types. We have natural media, we have artificial media. So media is generally defined as a medium which contains all the necessary elements which are required for the growth of a cell. So in the media or in the medium, we have all the like vitamins, supplements, everything for the growth of the cell, and even for the, if we are working on the tissue culture. So media is quite important. We have two types of media. We have natural medium as well as we have the artificial medium. So there are some another factors on which the effect on which the cell culture depends, like the pH. We have to maintain the pH also. So most normal mammalian cell lines grow well at a pH of 7.4. So we have to keep the pH at a normal level. Like some fibroblast cells will grow at a pH of 7 to 7.4. Okay, so we have to maintain the pH. Then again we have CO2, which is quite important in the cell culture labs. So the growth medium controls the pH of the culture and the buffer the cell lines in culture against changes in the pH. So CO2 is also quite important. I will share this PPT with all of you. So you have to read this PPT one or twice, and if you have any doubt or something is like that, you can share with me at any time. So now this is the general factors. The another thing which we have to keep in mind is the temperature during the cell culture lines. After it, this is the techniques. We have already done it, how to isolate the tissue, what is how to do dissection or disaggregation, culture after seeding into the culture vessel. So this is isolation of the tissues, the very first step, the very first step when we have to do the primary culture. So let me read it fast. So isolation of tissue, an attempt should be made to sterilize the site of the resection with 70% of alcohol, contaminate it so that there should not be any chance of contamination. So we have to use 70% alcohol to clean the surface first. Then we have to do this isolation of the tissue. Now this is the thing, that tissues can be isolated from mouse embryo, from chick embryo, from human biopsy material. So this is how to take the group of cells, how to take the tissues from the mouse embryo. They have just tell us the procedure. This is the whole procedure. Kindly read it by your own self. Like this is the experiment, how they are using, how they are just killing the mouse for taking a group of cells only. So this is the whole procedure. Kindly go to by your own self. So this is how the isolation of tissue takes place, how to do the dissection, and then what are the other steps which are there. Like we have to do the dissection first, then we have to do the mechanical disaggregation. After it, we have to do the enzymatic reaction also. So we have to do with trypsin along with EDTA to finally get the cell lines. So this is the whole chart. Kindly go through it with by your own self. So this is the whole steps how we are able to get the final cell line. But we have to isolate the tissue. We have to start with the dissection. Then after dissection we have to do mechanical disaggregation, and after it we have the enzymatic reactions which will take place, which leads to the cell lines. So this is primary cell culture. Again we have already discussed it. Kindly read it by your own self. Everything is easy. I hope you will get it. Then this is the enzymatic reaction. We have to do it by trypsin enzyme along with EDTA. Okay. After it we will able to get the final cell line. Then this is cryopreservation. We have already discussed it when we are keeping the tissues at a very low temperature, at a -degree Celsius temperature. This is known as cryopreservation. So basically what is cryopreservation? The best method for cryopreserving culture cell is storing them in the liquid nitrogen. So when we are storing them in liquid nitrogen, in complete medium in the presence of cryoprotective agents such as dimethyl sulfoxide. So in the presence of DMSO, we are able to this is the agent. Basically this is the cryopreservative agent. So in the presence of it, when we are able to store the cells at -degree Celsius, -196 degrees Celsius temperature, this is known as cryopreservation. So cryoprotective agents, which is DMSO, it reduce the freezing point of the medium and also allow a slower cooling rate, greatly reducing the risk of ice crystal formation, which can damage cells and cause cell death. So DMSO is known to facilitate the entry of organic molecules into the tissues. Handle reagents containing DMSO using equipment and practices appropriate for the hazard posed by such materials. So basically DMSO, basically cryopreservation is there when we want to store our cells, when we have to store our cells at minus degree Celsius temperature, when we have to preserve the cells. So there are some guidelines for the cryopreservation, how to do the freezing, how to take the sample outside. So like they are saying store the frozen cells below -70 degrees Celsius. Okay, so we have to keep these things in mind. You have to read it by your own self. This is again the protocol for cryopreservation. How to preserve basically the culture cells. These are the things which we have to which we have to keep in mind. We have to be sure that there should not be any contamination, there should not be any cross contamination either. We have to confirm of the species of the origin. We must know that from where we are taking those particular group of cells. Identification of the lineage to which cell belongs to, position of the cells, determination of whether the cell line is transformed or not. So all these things we have to keep in mind when we are working on the cell culture or cell lines. Why we are using this cell culture technique? What is the use of this cell culture technique? Yes, this technique is very recent in the field of cell biology. By using this technique, we are able to know some toxicity testing. We will able to know, or we will be able to do some toxicity testing. We are able to do cancer research, gene therapy, genetic engineering, virology. All these things are covered with the help of this cell culture technique. I think I speak quite fast today. I think I speak very fast. I have to change my video quality to 360. From where I have to change it, if anyone please let me know. I will change it definitely. I will share the PPT just after this lecture series. I will share right after it in your Telegram group, so that all of you will be able to see it by your own self. So if any one of you, please let me know how to change the video quality, from where we have to change it. Okay, I will try to. Okay. So yes, this is only the cell culture. Actually, I have to tell you only the basics of the cell culture, so that you will have to know a little bit knowledge about the cell culture only. Otherwise, today we have to discuss, or we have to do this thing, which is molecular biology techniques part 1 and part 2. I want to keep this on today, but some of you have maybe some exams. Now please let me know when you will be free on Sunday. Are you free on Sunday? Because I have to take your long session. I have to complete MBBT part 1 and part 2. So please let me know. Are you? I'm sorry. Please, I'm little bit fast today. I am fast because I have doubt on my net also, when it will break. I don't know. I'm not sure, na. So that is why I'm little bit fast. Moreover, I have to cover it today because we have not done it yesterday. So Megha, I'm really sorry for that one, please. But please let me know, are you free on tomorrow? Tomorrow is Sunday. I hope all of you will be free. So please let me know whether we will be able to take a 2 hour session. Are you free? May I allow to change the timing? Please let me know. I will share this PPT with all of you. Don't worry. Just let me know whether you are free on tomorrow so that I will change the timing. Whether you are free on tomorrow so that I will change the timing. Gurpreet, then please, please by your own self, please, you can watch the recorded session if you are not free, because I know Sunday all of you are free and I want to complete it little bit early so that you all will be free by 9 instead of just there along with me on 9 to 10. I just want to free all of you. So I will let you know the timing. Most probably I will take it after 4 p.m. Getting my point? So I will try to take it after 4 p.m. So is it okay to all of you? By 4 p.m. I think you all will be free from your daily activities, maybe like tomorrow is Sunday, so maybe you get late. So I think 4 p.m. is okay. 4 to 6 p.m. is fine. Yes, yes, this is available for download. I will share you the link and you can watch it at any time, no issues at all. So I think we will take it after lunch basically. After lunch, after a little bit of nap, 4 p.m. is I think it's okay. So I have to cover this topic which is there, which is MBBT techniques part 1 or part 2, or I will start recombinant technology. I will let you know, okay? So that we will be able to do it fast because tomorrow is Sunday, we have a holiday. Otherwise, we have only 1 day, 1 hour only, and I want to complete these techniques and everything. Like today is day 5 and we have just covered day 1 and day 2 only. Okay, so that is why we will have to be little bit fast. So I will meet you by 4 p.m. most probably. Okay, and I will share this PPT with all of you right now. If you want to do, then we will do a little bit about recombinant DNA technology now. Otherwise, we will do it. We will start it from tomorrow. If you want to do it, we will even start it right now. But I think cell culture is okay for today. Yes, yes, Alvia, I know you have some doubts. You can message me personally also. I will answer those queries. Just give me 1 or 2 minutes more. If you have some doubts, Alvia, then please share it in the Telegram or you can message me personally also. I will let you know. So I will take your class by 4 p.m. because I have to schedule the whole day tomorrow. So I will take it at 4 p.m. so that by 6 p.m. you all will be free. If you want to go somewhere, you can. So yes, tomorrow we will try to start this molecular biology techniques, or we will start recombinant technology. I hope you all will be aware of recombinant technology. Please let me know if you all are aware of it. Recombinant technology. Are you aware of this recombinant technology? This recombinant technology. Are you aware of this? Or you are new to recombinant technology? I will use this. Okay, because otherwise I have to start from very basic. That is why I am asking you all that please let me. Okay, someone is new. Okay, computer is no more. So we will start from very basics regarding recombinant technology. Please reply all of you. Uh, are you able to, have you done recombinant technology before also? So have you done recombinant technology before also? Please let me know. Okay, so we will start from the basics. Okay, no issues. I will start from the basics. Yes, yes, yes. This is recombinant RDN technology. Yes, Megha, yes. I confirm it already, Sandeep. We will take it from 4 p.m. That means I will take it after lunch only, not before it. So Neha, Dipti and Shumam, they are quite new to recombinant technology. Okay, then we will start from the very basics. Okay, okay, okay. Okay. So shall we start now or shall we have to start from tomorrow? I think this is 1 hour now. So I will share this PPT of cell culture right now with you so that you will be able to get everything which you want to know right after this class, and then we will start the recombinant DNA technology from tomorrow. Yes, yes, definitely. I will let you know about the timing. Yeah, yeah. Please, this is the one thing that I have to give you timing at least 5 to 6 hours before so that you all will be ready. Okay, okay. No issues. Let me make the schedule for today. Then I will send it in a group. Okay, but I will take your class after 4 p.m. because I have to take the class of biochemistry by 11 to 2 p.m. Yeah, 11 to 1 p.m. Then from 2 to 4 p.m. I have to take the class of genetics. So after it I will take maybe from 5 or maybe from 4:30. So that is I will send you the schedule, no issues. And one more thing is that today's Telegram is not working in my previous session also. So sometimes I'm not able to share the link on Telegram. I don't know why it is not working today. So I request all of you to please subscribe to my channel so that you will be able to get the notification when I will be live, and it will be helpful to all of you and easy to me also. Okay. So that's all for today. I hope you will be able to get it today because from the past 2 3 days we are not able to you know do the study as we have to do, and even I am not satisfied if I am not able to do my best. I want that my students will be there throughout with me and they will not feel bad or disturbed. So I'm really sorry for it, but yes, we will tomorrow we will meet. Okay. So take care of yourself. Okay, okay. So there was an update on. Okay, okay. Sure. Yes, that is why the Telegram was not working on my MBBT, not on my biochemistry batch from 4 to 6. So I feel very bad at that time. So that's all from my side. Please and take care of yourself. Good night. After finishing this video, I will send this cell culture techniques to your to your Telegram group. So thank you all. Take care. Take care. Thank you, Dipti. Thank you all. Please take care of yourself. Bye bye. Good night. Enjoy your day. The day is over, but still we have whole night. I know all of you are using your phones or something like that. So take care of yourself. Good night. Bye bye. And please subscribe to my channel. It's my request to all of you. Yes, yes. Good night to all. Good night. Good night to all. Take care of yourself. Yes, thank you to all of you. Thank you all for the cooperation. Take care. Bye bye. Bye bye. Take care. Good night. Okay, okay. I will check it. I will check it. Just after after just closing the laptop, then I will check it. Yes, yes, you can download it. Even I will share the link with you. You can click it and at any time and you can watch it at any time. No issues at all. Instead of download, because when you will do the download, it will your memory gets full. So rather than save it only, and when you want to revise it, just click on the link and you will able to get it. Okay, take care of all of you. Bye bye.